Assalamu alaikum dear students, I hope you all are good. Today we are starting with Emerson's red drop experiment. So today in this experiment we are going to start the wavelengths of the light at which the plants show the maximum photosynthetic yield. First of all, there is Emerson's red drop experiment, and in this it was conducted by Emerson in 1948. It's called as Emerson's red drop effect or Emerson's red drop experiment. In this, what he observed, when plants are given a wavelength of above 680 nanometers, plant showed a sharp decrease in quantum yield. Let me tell you what is quantum yield. It is the number of oxygen molecules released per quantum of light absorbed. Number of oxygen molecules released per quantum of light absorbed means it shows the rate of the photosynthesis. As a result, what Emerson concluded, he concluded that there was a drop in the rate of photosynthesis when wavelengths above the wavelength 680 nanometer were given to the plants. Above the 680 nanometer, it showed a drop in the it showed a drop or decrease in the quantum yield, you can say. Fine, this is Emerson's red drop experiment. This is not in your syllabus, but to make the things clear, I have chosen these experiments. Now there was Emerson's enhancement effect. When Emerson observed that when wavelengths of light 680 nanometer above were given to plants, there was a decrease in the photosynthetic rate. Next, he conducted another experiment that is called as Emerson's enhancement effect. Then Emerson, what he did, he combined two wavelengths. Those are far red and red light. Far red is above 680, that is 700 nanometer, and red light is 680 nanometer. So when Emerson combined light of wavelength 700 nanometer and 680 nanometer, he showed that the rate of photosynthesis was more than double, more than the sum of the rates of the photosynthesis at far red and red light. What he observed, if he took the individual rates at far red, that is 700 nanometer, and red light, that is 680 nanometer, it was more than that if he combined both far red and red light. So here is the slide I'm showing you. Again this axis we have plotted the rate of photosynthesis, and against this we have plotted the wavelength of the light given to the plant. So if there is 680 nanometer red light is given to the plant, there is a bit increase in the rate of photosynthesis, here you can see. But if the far red light is given, that means the wavelength above 680 nanometer, that is far red light 700 nanometer, there was a decrease in the quantum yield or decrease in the rate of the photosynthesis, here you can see. But then Emerson conducted an experiment that's called as Emerson's enhancement effect. In this experiment, he combined both. He gave the wavelengths both 680 and 700 to the plants. Then he saw what there was a maximum increase in the rate of the photosynthesis. More than these, if these two are combined, more than these, there was the rate of photosynthesis. So I hope I am clear. This is Emerson's red drop experiment. When Emerson gave the wavelength above 680 nanometer, there was a sharp decrease in the rate of the photosynthesis or the quantum yield, and at red there is a slightly increase in the wavelength of this rate of the photosynthesis, and when he combined both, there was a sharp increase in the rate of the photosynthesis. I hope I am clear to you guys. Next, we're already discussing the photosynthetic pigments or pigment systems or photosystems. Now let me tell you these pigment systems. I have already told you the structure. There is antennae complex, there is the reaction center. So now these photosystems in plants are of two types. There is photosystem 1 or P700. Let me tell you in this what happens. It is driven by a light of wavelength 700 nanometer. Why this is called as P700? Because it is driven by a light of wavelength 700 nanometer. Fine, because the chlorophyll molecule present in the reaction center of this photosystem first absorbs the light of wavelength 700 nanometer. That is why it is called as P700. P stands here for pigment and 700 means it absorbs the light at 700 nanometer. Fine. In higher plants, if you see the reaction center, I am telling you the structure of this. In higher plants, if you see the reaction center contains chlorophyll molecule which absorbs the light of wavelength 700 nanometer, and that is chlorophyll A molecule. And it has about 100 core antennae chlorophylls, और जो इसका antennae complex है, in that it has approximately 100 chlorophyll molecules. And there are proteins also. I have told you that in antennae complex the pigment is bound to the proteins. There are two proteins which are PSA-A and PSA-B. Fine. I hope this is clear. Now if we come to pigment system second or photosystem second, that is P680. Why it's called as P680? It's understood because it is driven by light of wavelength 680 nanometer. Fine. Means the reaction center of this contains a chlorophyll molecule which absorbs the light of wavelength 680 nanometer. That is also a chlorophyll A molecule. Fine. In higher plants, chlorophyll molecules in reaction center are associated with two membrane proteins. अब जो इसमें proteins होते हैं, those are D1 and D2. In that I have told you those are PSA A and PSA B. Please remember in photosystem 1. In photosystem second we have D1 and D2. So let me tell you, you must be wondering that in the reaction center of both the photosystems there is chlorophyll A molecules. Then why this absorbs the light of wavelength 700 nanometer in chlorophyll, this pigment system first, and 700 in chlorophyll, this pigment system second. So you must be wondering that the chlorophyll A molecule is same. It is because the different proteins are associated with the pigment system. It is not because of the chlorophyll molecule. It is because of the different proteins that are bound to the pigments in the reaction centers of the both pigment system first and pigment system second, that they absorb in the different wavelengths. I hope I am clear here. So let me tell you before coming to the distribution कि ये कहाँ पे होते हैं pigment. अब आप बोलोगे thylakoid में होते हैं, but where in the thylakoids pigment system first and pigment system second occur. So let me tell you, first of all, you know the structure of the chloroplast. These are the thylakoids, 1, 2, 3, 4. These are thylakoids stacked upon one another, and these form a grana. So the thylakoids, individual thylakoids in this grana are called as grana thylakoids. Now the thylakoids which lie in the stroma part are called as stroma lamellae or stroma thylakoids. These are also called thylakoids, but these lie in the stroma. Hence these are called as stroma lamellae or stroma thylakoids. Fine. So two types of thylakoids we have, grana thylakoids and stroma thylakoids. So this was my point. Now coming to the next, distribution of pigment system first and pigment system second. So coming to the distribution, you must know another concept. So this is a grana thylakoid, this is a stroma thylakoid. You see here the dark region, this dark region till here dark regions of these are called non-appressed thylakoids, क्योंकि ये appress नहीं हुए हैं. These are non-appressed thylakoids. And here non-appressed thylakoids are these dark regions. And here the light regions which are stacked upon one another, these are appressed, थोड़ा press हुए हैं एक दूसरे के साथ, and these are called as appressed membranes or appressed thylakoids. जो light regions हैं, these are the appressed membranes. So coming to the distribution, this was the point was made to be clear. So let me tell you, pigment system first is found exclusively in non-appressed membranes of grana thylakoids and stroma thylakoids. जो pigment system first है वो कहाँ पे होता है? It is present in non-appressed membranes. Here you can see जो non-appressed membranes है here. pigment system first, we are talking about the pigment system first. It occurs here in these dark regions, non-appressed parts of these granathylakoids and stromathylakoids. So coming to the second pigment system second, it is predominantly found in the appressed membrane, appressed regions of granathylakoids. Appressed regions are only present in granathylakoids. So here is the pigment system second. I hope I am clear. So let me recapitulate a bit. We studied the Emerson's red drop experiment, in which we found that when Emerson gave red light, there was a slight increase in the rate of the photosynthesis. But when he increased the wavelength of the light, fine, so he gave 700, means about 680, it was for red light, and it showed a drop in the rate of photosynthesis or quantum yield. It was red drop, it's called as Emerson's red drop effect. And then what he did, he combined both these wavelengths and gave it to the plant, and he showed there was increase in the photosynthesis more than these two. If we combine these two individually, more than these, there is an effect in the rate of photosynthesis. So it's called as Emerson's enhancement effect. So I am clear to you. I have already told you there are two photosynthesis, photosystem 1 and photosystem second. Photosystem 1 absorbs in at 700 nanometer, and photosystem second absorbs at 680 nanometer. And I told you where are these found, these pigment system first and pigment system second. So I hope this is clear. If there is anything, please let me know in the comments. Thank you. Stay blessed, stay safe.